let's start. So, any questions about selection? Everybody understand what selection is? Has some idea? Okay, what about drift? Everybody understands drift? No? So if you have specific questions, ask. Because it's a very important thing, and this is the sort of first time we are introducing in this course. So at least you have some idea. Tell me just in 2 lines, what is the most important thing you learned about drift so far? Anybody? Yes, you. Nothing you learned, okay. What about bench? Third person? What did you learn about the drift? Which was like something which will stick after you leave this course?
It's a random change in allele frequency due to sampling error.
Yeah.
That's genetic drift.
But I think you knew already before. So what did I teach you? Let me ask a slightly different way. If I have a population, so I was asking some of you in the lunchtime also, if I have a population in which I have 99 individuals of same type, and there's one special one, okay, and these 99 are, and there's one special one, they have same fitness, okay, so it's a neutral. The fitness of the white ones and the blue ones is same. So they evolve in some ways, there's some rules and all we can cook up, no selection. Do you think this can happen? What do you think? Yes? Why? This is weird, no? I mean, one rare mutant, do you think all 100 can become the blue one? Given sufficient time? Yes? Yes. Yeah? Why is that? Just by chance. So there's no selection. I'm not selecting. Sorry? No, in the selection, it will happen. It's not even in the... Without selection, it can happen. Yeah. This is without selection I'm asking. Okay? No mutation, no selection. Okay? Still, it's possible that all of them become blue one. That's kind of weird if you just sort of think, first time when you see it. But then you get used to it. Yeah.
We saw before the lunch that even if S tends to 0, which is like no selection, still the probability was 1 by n, which is not 0. So it means there is slight probability that it will get fixed.
That's precisely what I'm asking. It's neutral. So the probability that this would happen. That means if I did, 1 million numerical experiments, let's say, with these 100 individuals, what's the chance, in how many experiments would I see that all have become blue? I did 1 million experiments, numerical, let's say, okay, with some rules I have specified, let's say, and there is 100 individuals. In how many experiments would I see that all have become blue? Could you say that? It is haploid. I don't think it matters here very much. Yeah, so I did 1 million experiments. In how many experiments would I see?
1% of 1 million. 1% of 1 million.
How much is that? 10 to 4, just say that. That's all I wanted to hear. Okay, so the thing is that, in a neutral mutant, this can also spread, and the chance is 1 by n. I'll show this to you in another way when I start, introduce to you coalescent theory, or coalescent, way of doing these things. Before that, I want to do a couple of more things. So let's turn off the drift for a second because we have had enough of it for a while. I want to do 2 things. One is mutation because it creates variation. And secondly, I want to talk a little bit about recombination. Just tell me one thing. Did you hear about LD or linkage disequilibrium so far in these lectures? I have been asleep sometimes. Not at all? Is there anybody who doesn't want to know about LD? Who doesn't want to know? Nobody, right? Okay, so I'll do it. Okay, so let's start with you guys, which is that you help me in setting up the equation when I have only mutation. So no drift, No selection, only mutation. How do I go about it? I mean, I've been saying please write equations, so I expect you to write it. So please help me, how will you do it? So I'll tell you the model, only mutations. So I have A1 and A2 as before. So let's say A1 becomes A2 with some probability mu, and back mutation happens with probability nu. Okay? And I want to know how does the frequency of these two changes with time. It's only mutation. How do we write these equations again? Let's go through it a little bit. So what can happen? So I want to know what happens tomorrow. And the only possibility is that A1 can become A2 or A2 can become A1 with some chance. There's some chance of that. So if I want to count how many are there tomorrow, what kind of individuals are going to contribute to it? Can X1 contribute to X1 tomorrow? A1s can contribute to A1 tomorrow, yes or no? When can they do? Provided they don't change. Very nice. So what's the chance of that? 1 minus mu. So with mu, they change. A1 becomes A2 with probability mu. It remains A1. Let me expand this part. With 1 minus mu, it remains same. Right? Similarly, A2 becomes A1, probability nu, and remains same with 1 minus nu. Right? So very nice. You guys said that X1 would contribute provided A1s don't mutate this term, right? What else can happen? Who else can contribute to A1? A2 becomes A1. What's the term I should be writing? So there are x2 of them. Fraction is x2 and nu. Oh wow, all of you are very good. Can you finish this last line? Just write exactly, this is very similar. Test yourself. I don't need to test you always. You should test yourself. Can you do it? Dan, can you call out the equation, please? 1 minus? Yeah. Nu is the, what you said? Yeah. Yeah. People agree? And does it make sense? Is it correct? How do you check your equations are correct? Yeah. Because x1 plus x2 should remain 1 at all times. So this should be 1. And let's check. This plus this plus this plus this is indeed 1. So he has got the right equation. Right? Okay. So very good. What's the equilibrium of this population, of this in this case? Again, equilibrium, remember, when things don't change in time. So therefore, x1t is same as x1 at t plus 1, x2 at t is same as x2 at t plus 1. So therefore, what happens at large times? What's the equilibrium? Are they equilibria or equilibrium? At large times, what's the frequency? Let's hear from somebody else. What do you want to say? Yeah, please solve. Solve one of the equations, that's enough. Because x2 is same as 1 minus x1. Use that. Right? Yeah? If you're done, tell me how x1 changes as a function of time. Those who are done with the equilibrium. Would you like to tell me? Yeah, done. Okay. How about you? Your neighbor. Okay. Those who are done with equilibria, please solve and tell us quickly how much x1 changes in the function of time. Otherwise, you just do equilibria. That's enough for now. Feel free to chat with your neighbors, discuss, that's okay. Are you done? Yeah, tell me. X1 you're saying? Is it new or new? Yeah. Is this new or mu? Is this correct? Huh? Okay. 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 So I think you can all do it because it's a simple linear equation. Sorry? No. Mu and nu are not, mu plus nu is not 1. The denominator should be mu plus nu. Is that what you meant? This is correct as I yes. Okay? This formula makes sense. Let's set mu and nu equal to nu, let's say equal to half. So A1 can become A2 with probability half, A2 can become A1 with probability half. Without doing the calculation, what would you have expected? So there is no asymmetry, like A1 can become A2, the same chance as A2 can become A1. So you take this blue balls, put in this one, it has red, and you take the same amount, with the same chance, take the red ones and put in blue ones, and you just keep mixing them. So what do you think is the frequency of x1, the frequency x1 going to be? Without looking at the formula. Huh? What would you have expected? Half, right? Makes sense? And that's what this formula is also telling you. Okay? Okay? And therefore, the one which has a higher mutation rate, suppose nu was very small compared to mu, which frequency would be higher? Nu is 0.1, mu is 0.9. Or let's say nu is 0.1 and mu is 0.8. Which one do you expect would have a higher frequency? A2? Because things are converting more often there, right? But it's a very tiny small formula which tells you exactly how much would it be. So again, verbally you know which one would be higher or smaller. These little two-line calculations tell you how much would they be. So once again, they are worth doing, so please do them. Okay? So this was simple. Let's do something complicated now. So I want to bring in recombination. So if you understood this kind of equation, it should be relatively easy to write the case of recombination. So that's slightly more involved. So I'll go a bit slowly. Okay, so can I continue some sort of same model for understanding to include recombination, or should I make any change? I have been considering a haploid population, and I want to understand what recombination does. How do frequencies change under recombination? So what should I do? What kind of population should I be considering? Diploid, right? Okay, so I'm thinking of now diploid populations. And when we say this, we have just A1 and A2, these are called one locus problem. That means you are not considering whole genome or something, you're just saying, I'm just looking at this portion, I'm going to call it A1. But there's an A1, and adjacent to it, there's another one, two loci, and I'm considering two loci. That's what I need for recombination, because it has to break and swap the parts. So this is a diploid two locus problem. Okay, so that at least that much I need if I want to talk about recombination. So I'm going to introduce the following. So I have these two loci. Okay. So this locus I'm going to use letter A, and for this one I'm going to use letter B. Okay. So biologists, tell me how many zygotes can I form? How many zygotes? Zygotes, you guys know better than what zygote than me. What are zygotes? So again, it's biallelic, meaning I can have only A1 and A2 here, okay? And here I can have B1 and B2. What are the possible zygotes you can make? Okay, let's talk about gametes if you like. What are the gametes we can form? 4. What are they? A1, B1. Okay, let's stick with gametes then. So these are the gametes. And I want to write down the equation for the frequency of these gametes. Only recombination, no drift, no selection, no mutation, just recombination. What does it do? Okay. So frequencies are x1, x2. So just like with cytomutation, that I want to know x1 in the next generation, how do I get it? Somebody should mutate, A1 should mutate, or A2 should not mutate, etc. That sort of thing I wrote down, right? I want to do the same thing for these frequencies. So tell me, which gametes should recombine so that they contribute to A1, B1? Suppose, let's see the meaning of recombination first. Suppose I have this gamete and A1, B2. By recombination, I mean the following, that this breaks and I swap the material so that I get A1 swapping. Right? With some probability C, or let's call it R, recombination rate. Suppose this happens. Did the frequency change? No change in frequency, right? So what kind of recombination events would contribute to the frequency of A1, B1? Which kind of recombination event should occur? A1, B2 and A2, B1? Is it correct? Okay, so let's write down that contribution. So x1 at time t1, if there's no recombination, what happens? It remains same, nothing else. I mean, that's the sort of thing you saw it here. If mu and u were absent, it remains the same, no? So there is that term. Plus, with probability R, these two should come together. The frequencies is x2 and x3. So now you are seeing a very different kind of equations. It's not just x1 or x2. It's not linear anymore. Things are going to become non-linear. Recombination is very difficult to handle even theoretically. x2, x3. Is this much okay with you? No, so I could do it that way. The way I could have done with mutations, I could do the same way, no? Like no mutations, so therefore the frequency should remain the same. Plus the changes.
When you talk of recombination, it happens for any two, right?
Yeah.
So here, the one A1, B1.
So this one would have recombinations of the kind that I erased. This events. No, A1, which one? A1, sorry, say that again.
A1, B1 with A2, B2.
A2, B2, yes. Yeah, but yeah, I haven't done that. I'm coming to that particular, that term precisely. What's the frequency of those two? No, that will reduce the frequency. Minus R, right? Yeah, so that's what exactly I was coming to. These recombination events increase the frequency, just like that mutation case, but it's slightly more complicated, right? Minus A1, B1, A2, B2. When they recombine, what happens? Do they contribute to A1, B1? Of course not. They're reducing the frequency. What are the terms? X1, X4. Is this okay with you guys? This is slightly complicated, but it will bring out a very important thing. That's why I wanted to go through this. Yeah. So then it changes, yeah.
So XP can't remain x1 of t, right? There should be 1 minus r. Because that is the proportion.
This contains all those guys which recombine and did not change things. Just like for that one.
There again we use 1 minus mu, right?
But you can rewrite, right? Analogous to this. Okay? Is this okay? If this is okay, tell me x2. Let's fill up this table because then it will be easy. Which one should recombine here? Yeah. A2, B2. Right, what else? Okay, I'll just fill it up. I think you guys know it. But see if you agree. Okay. Can you tell me x to t1, t plus 1, please? Which ones for x2? I'll get a1, b2 from a1, b1 and a2, b2. x1, x2? x1, x4. Yeah. And minus? Yeah. Very good. Let's just moronically do it because it's worth the effort. Next one. Okay. x2, x3. Huh? And very good. You guys are somehow better at recombining populations. Wow. Okay, I'm impressed. Now this looks pretty complicated. But what is this object? Same combination, same combination, same combination, no? Up to a sign. Right? What is this object? Anybody recognizes it? Determinant? Huh? ऐसे ही कुछ भी something it has to be. Don't throw words, man. Tell me. You are almost there, but you have to say more. Okay, I'll complete the sentence. Oh.
Basically, you are saying that kind of not recombining in this because it's become.
I'm asking, what's the meaning of this term? Do you think this same thing is coming in all the four equations? X2, x3 minus x1, x4. Is there meaning to this combination? Okay, I'll tell the answer. Just one sec. There's a meaning to it. It's a name for it. It's called linkage disequilibrium. It's a very famous quantity. Yeah. Only, oh, sorry. I'll ask you another question. Yeah, let me just write it. I'll come back to you. Yes.
Does the first term implicitly considers the recombination between A1, B1 and A2, B1
The ones that did not change?
after recombination?
Yeah. So those ones are included first term. But you have to work it out. Okay? Okay, so this looks, complicated. It's pretty complicated. And I don't know what's the meaning of it and all. So let's just do one more step to understand the meaning of it. Okay? Let's rewrite this object. Because x3 is 1 minus x1 minus x2 minus x4, right? Right? All these frequencies should add up to 1. So, okay? I'm rewriting it because I want to tell you what the meaning of this object is. Okay? And after something, it equals, okay? So x1 we know, it's a frequency of A1, B1. Oops. I hit wrong here, okay. What are these two objects? Did I do it right? Oh. Sorry? Yeah, I think I missed something here. Could you check quickly, possible? This is X1, is it correct? No. Okay, sorry. Should I check? I think what I have in my notes is something like this. Okay, I'll check this point, but it turns out, let's see it. Okay, what's the frequency?
Ma'am, that is x2 minus, I guess, that first term.
It should be X2?
x2, yeah.
If I'm writing this, then it should be X2, yeah. Oh, no, but you're right. Yes, that's a linear term. Oh, very good. Thanks. Oh, wonderful. Okay. Yeah. So then you're right. Okay. So this one we know, X2. Over there we wrote A1, B2. What is X1 plus X2? Does that have a meaning? What will it give me? Frequency. You saw this in the Hardy-Weinberg discussions. So that's why I'm asking you instead of telling you. Is the frequency of? Frequency of? No, we'll come to that. Just tell me X1 plus X2 gives the frequency of what? A1. Because I don't, it's saying second locus, I don't care whether it's B1 or B2. It just gives the frequency of A1. You saw this, right? And similarly, X2 plus X4 is frequency B2. Right? So linkage disequilibrium is saying D is the frequency of the joint object A1, B2 minus the frequency of them separately. What does that mean? Is it clear what you said? People haven't seen LD at all in your entire life. How can you be so old and not know LD? Sorry, I didn't mean to say that. So what I mean here is, see, A1, B2, when is it? So what does recombination do? Recombination is breaking these associations. So A1, B2 is coming together, my friend, like that, right? Recombination is going to break these associations, okay? So when it breaks the associations, what you will not see is this combination, the combined object. You will see A1 separately, B2 separately. It will have A1, sometimes B1, sometimes B2 with it, right? So if you see A1, B2 together, minus A1, whether it comes with B1 or B2, times this frequency of B2, whether it came with A1 or A2, this difference, this random associations, the measure of the random associations versus when they are associated with each other, that measure is called linkage disequilibrium. It's the linkage, a physical linkage. Okay? So asexuals, do they always remain linked? Asexual objects. There's no recombination, right? So the linkage disequilibrium does not change. But I'll give you the answer here. Actually, there's some physicist here. Physicist, do you recognize this object? This kind of thing. Correlation. There's another word which one can use. It's saying how things are associated or correlated. That's what LD or linkage disequilibrium measures. Okay? So A1, B2 together minus the frequency when A1 came with either B1 or B2, I don't care. Frequency of B2, whether it came with A1 or A2, I don't care. That difference, if they were random, it would have been 0, but it's not usually. Okay? So that measure is called linkage disequilibrium. And if I plug in, I'll not do this further, but if you just manipulate these equations using this equation, it turns out that D of t plus 1 goes as, it obeys this equation. That is, as time goes on, linkage disequilibrium, what do you think? Is it decreasing, increasing, or same? Decreasing. When I have recombination, it's reduced from the previous ones. That's what recombination does. It decreases over a time scale, which is of the order of 1 by r. Okay, so if they have r is 0, that means it's an asexual population. LD remains same. But with recombination, you break LD. LD decreases. The things which are coming, if you are, my friend or something, it just came by chance. It's not you want to be specially with me. Okay? So that kind of chance associations, random association, versus when they are actually together. That difference is what LD measures. So recombination is the one which decreases it. Okay? And the reason I wanted to bring is because you will, I think, see it in later on in some of the talks perhaps. So we have been doing all these single locus models. Nothing is single locus, right? So you guys have been worrying about, oh, but genome is so long and this and that. Why am I clubbing everything, putting it in just this big stretch and just call it A1? This big stretch, just call it B1. What's that, right? But if your population is sufficiently recombining, then because its associations break down, one locus results are a pretty good approximation. LD decays very fast. Is that sort of clear? It's not a very simple concept. You have to think a little bit more about this. Okay? So I'm saying I have this genome just with two loci, this one, and because the recombination break down, but sometimes this guy will come with it, but it just happens to be, by chance it happens to be. By chance here I don't mean drift. It's just sometimes B2 comes, sometimes B1 comes, and so on. But these associations will break down pretty soon if the population is recombining. And in those cases, it's good approximation to use these kind of results or the morning results that I told you about drift. So one locus results are good approximations when the population is recombining. When they are asexual populations, then you have to worry. There is LD and you have to take care of it. Okay, since you are hearing LD for the first time in your life, so it'll take a while. So think about it. If it's not clear, ask me next time. Okay, so I want to do one model. I wanted to now introduce to you a bigger model, and it's a sort of a prototype of whatever we do. Lots of things are done, and it's called a Wright-Fisher process. How many of you heard of it? Wright-Fisher model, Wright-Fisher process? Okay, let's do it because that will allow me also to introduce coalescent. So let's do that. So I'm going to put now everything except migration, the limitation, the selection, the drift. I'm going to give you a model. We're not going to solve it or anything. What you can do, if you have time, is to simulate it. Okay? So let me tell you what this model is. Nobody has ever seen in your textbooks Wright-Fisher process. You might not know it, and what it is, but have you ever seen this phrase? We simulated a population using Wright-Fisher process. How many of you have seen that? Seen it, no? Rest of you have not seen. Okay. No, I did a little calculation which I didn't show. Okay.
Once you said intuitively, you said it. Can you just repeat it?
Because recombination will break those associations, no? That's what our terms were coming.
So even saying something like even if it is happening randomly, due to recombination, that will also reduce.
The recombination will make things more randomly associated. If there's no recombination, you stay with your friend. So here I didn't consider selection as something. But what if there's selection also with recombination? What if there's drift also? Then what happens? I don't know. Yeah.
Can selection make R 0?
No, R is what we put in. R is the chance with which recombination occurs. Probability of recombination. Oh, yeah, right. There's asexuals, yes. For asexuals?
It's sexual and recombination.
Oh, okay, actually, for me, sexual means recombining. So what I mean here is just genetic mixing, in which you break the parts and swap. That's what I mean here.
What I'm asking is R, can selection control R?
I don't know.
Because only then we can say that a block will remain a block.
No, I'm saying it's an intrinsic property which came about for whatever reason. So that comes in the category of this evolutionary genetic systems problem. So why is the R 0.1 whatever, I don't know, because of selection or because of drift or whatever it is, we don't know right now. Maybe it's shaped by selection, I don't know this. It's a very complicated topic, I just don't know properly. Yeah.
How fast does LD decay like?
Yeah, you should be able to tell me that. This is right there, no, come on. Yeah.
We know about hitchhiking. So does recombination reduce hitchhiking? Because we see when from this relation.
So when we are talking about hitchhiking, we are thinking of a selected variant. So I think most people don't know what this is, but I'll just think of, I'm just telling you the standard result, which is that you have a special variant, okay, and others are neutral sites. And the selected variant is going to fixation, right? That's what you have in mind. So what hitchhiking, what will happen is that the neutral diversity reduces. If there's recombination, then you think it will be less or more? So when there is recombination is less, because it drags things along with it. That's the meaning of linkage. It drags things along with it. So then diversity, nucleotide diversity reduces. Yeah, that's what, yeah. So recombination keeps it same or close by. Okay. Okay. I'm going to introduce this model. Simulate it, please. Okay. It's called Wright-Fisher process. So I'll just tell you the rules of the game. It's just a game, okay, which you can sort of put in your computer. So what I'm thinking of is are n individuals, haploids, and they're asexuals. And let's say again one locus, but you can easily generalize it, okay? And there's selection. So I have these two types. Let me call it zeros and ones. Okay, let's do A1 and A2, it's okay. Fitness of A1 is 1 plus S, or I don't know what I wrote earlier. Okay, 1 minus S. Okay, and then there's mutation. So A1 and A2 can convert to each other with some probability. And of course, there's drift because I'm dealing with a finite population. Okay. So I have n individuals, yes. From what I understand is that you have this entire genome and some things are happening. But I don't want to see all that thing. I just coarse grain it. That means I zoom out and say this thing is wild type. And everybody else who is different is a mutant, which I'm calling so this whole chunk, I'm replacing it with A1. And this whole chunk with A2. That's what I mean. So the whole chunk I'm replacing with by this by just a single number, by single type. Okay. So that's where the development of population genetics was in the last century, and still took almost 50 years. And when you bring in more loci, you have to worry about those kind of effects, and it's quite difficult to do theory. I mean, of course, simulations are also difficult, experiments are difficult, but that's where the real action is. When you have lots of loci under selection simultaneously. That's really the research area. But it is very challenging, and I guess that's very exciting too. Okay, so let's say n is 5. So I have A1, so at t equal to 0. Let's call it A2. I'm writing this, but there's no real space. This is not some spatially structured populations. There's just 5 individuals. You can write them the way you like. So it's my first individual, second, third, fourth, and fifth. Okay, so I have 3 A1s for some reason and 2 A2. Okay, so I said you evolve the population for some rules. So I'm giving you a specific set of rules. So this is how you do in this model. So A1 will produce its offspring, A2 will produce some offspring because A1 has some fitness. Okay, let's call it W1 because it's more general. Okay, A1 has fitness, let's say higher, some value, right? Okay, so in the next generation, each of them will reproduce. Let's say, first of all, there's a reproduction step. So I said W1 and W2 are the number of offspring produced per individual per generation. That was the meaning of this Wrightian fitness, right? So let's say A1 is, W1 is, let's say 3, W2 is 1. Okay, let's call it 2. Okay, so on an average, A1 produces 4 offspring. Right? But it's easier to sometimes work with models in which population is exactly fixed. So I started with 5 guys. I don't want to make it such an expanding population. Okay, so the rates are such that things remain more or less at the same value. So therefore, I have now lots of them. So what should I do? Lots of these guys have to die, no? Because I have food only for 5 guys. So what do you think? Who should die? How should I decide? Hint, I'll give it to selection. Reproduction has happened, right? So selection, yes. But selection is sort of, okay, this is reproduction step, fine. Okay, so how should I kill some of these guys? No, say it's a more concrete thing, no? Randomly means what? I toss a coin and ask, kill this guy, mafia guys. No. Toss a coin? Half. Nah, then? Yeah. So I'm asking, should I kill this guy or not? What do you think is a reasonable way of doing it? Forget Mr. Wright and Mr. Fisher. Equally likely? Equally likely? Exactly same, yeah. I mean, don't you think it should depend upon the fitness, no? Huh? So you think that these guys are more likely to survive? Yeah, so something like that you could try. There are various ways of doing it. So I'll tell you one way. So you can do this way and try to kill, let's say with equally likelihood, and see how the population changes over time. Okay, I'll tell you one way of doing it, which is that you say that this is going to produce an offspring with a probability which is proportional to its fitness. Since this is fitter, it's more likely to have a baby. Sounds reasonable or not? Right? So I'll throw some random numbers in the computer, okay? And the chance that it produces is proportional to W1. The chance this does is proportional to W2 and so on. You'll have to write this bit of program sitting with your, friends who are comfortable with it. So, but that's basically what I mean here by putting selection in. So, it so happens I got, is this possible? This genealogy? Possible? Sure. Then I can put mutations. So then A1 can become A2, maybe it didn't, maybe it didn't, maybe it did. So that's how I form my new generation. Oops, sorry. So 3 A1s have become 4 A1s. Happens to be. So this is one stochastic trajectory. If I had thrown different random numbers, I would have gotten something else. How about this one? Let's forget mutations for a second. So I continue this. No mutations. It's like a t equal to maybe 1000. Is that possible? Yes? Why? What happened to A2? Very nice, very good. This is possible? Is there a drift in this? No drift? No, there's no drift, no? So people are doing this, some are doing this, some are doing How many say there is no drift? Raise your hands, like me. Who is with me? No drift. Raise hands, guys. Who says no drift? Who's with me? No drift. Okay, we are all losers. Because there is drift. Drift, what is drift? Drift comes when I have a finite population. Right? I have a finite population, n. कितना बड़ा बड़ा लिखा ना? N, big N. When I have a finite population, I have drift. Drift is stochasticity. I haven't shown it to you, but you can show that these kind of fluctuations happen when n is finite. If n is infinity, fluctuations go away, there's no drift. You're back to what we started with. Okay? So you will not see such a thing. A2 will not see. So this is one thing which is very different qualitatively. If I have a finite population, the bad guy can fix, which I said in the morning also, and infinite population, that never happens. If you have infinitely large population, A2 can never fix, whereas the bad guy can fix with a small probability, right? Okay? Is this sort of a little bit clear what drift can do? The only thing at least you should take home is the drift means fixation. Fixations can happen if there are no mutations and there's drift, then one of the types will eventually fix. Is this okay? Drift and selection, one of them will fix. Clear to everybody? Because I'm going to ask you something now. Yes. Right. Yeah, but it's still possible, no? Even if they were equal fitness. Okay, this is what I started with today in the second lecture. Even if they had equal fitness, do you think this is possible? Okay, is this okay with everybody? Let's go ahead with, I'll see the coalescent thing, we'll see how much time is left, yeah. If there are no mutations, yes. One of them, by just by fluctuations, only one of them will survive. There's a chance. There's a chance for that. It's not definite. And that's what we calculated. I mean, I give you the answer, sorry. Right? Okay? So if this is clear to everyone, and we have 20 minutes, and it's time for a problem. I want you to use these two concepts. First, I introduced to you mutations. I said, we started with that, right? So if A1 can become A2, and A2 can become A1 with equal probability, what was the, without drift, what was the frequency of X1? Look at your notes. No, equal probability. A1 can become A2 with half, A2 can become A1 with equal probability. How much is the frequency? Half. You have to remember it, right? This is something you said intuitively understand. Isn't it? Right? Okay. So I want you to tell me the following. I have this population which is evolving in this using this Wright-Fisher process, but there's no selection. There's only drift and mutation. So W1 and W2 are equal. Say 1. Fitness is 1. Okay? So they are just neutral variants. So there is drift and also there's mutation with equal probability. Okay? So I ran the simulation. And what will happen? What do you think will happen at large times? See, he was asking that, will one of them will get fixed. That is selection and drift. I'm asking a different question now. I'm asking mutation plus drift, no selection. What do you think will happen to this population? No, so no, let's give me configuration. Would I see all A2s? What will I see? Can you tell me? You just came. Okay, anybody else? Yeah. Sorry? Mutations and drift, yeah. Please take a mic, I can't hear you.
So initially, if drift is acting on, then one population will be a bit more than, but since mutation is also acting simultaneously, then it will be equal proportion of both, because the rates of forward and reverse are equal.
No, but I mean, I'm not talking about average. I'm saying I started with 3 A1s and 2 A2s, and all that is happening is that these guys are equally probability selected. So this has the same chance of having offspring as this one. Okay? That's what the meaning of neutral is. And but A1 can become A2 and A2 can become A1. So do you think this is possible at very long times? No. What is, can you give me one configuration?
So if it is like neutral, then the ratio will remain same, no?
No. Okay. So all A1s is possible? At that instant, yes, it's possible, yeah? So after that, I keep looking, today, tomorrow, and day after. Is it possible that all will become A1 and tomorrow I come, all are still A1 and so on? That's not possible because of mutations. So if I count the number of A1s that I see today, I come tomorrow, again I count the number of A1s and so on, it's going to be a different number. That means the frequency or the number of A1s is a random variable, right? I'll see different ones. So what I do is, I do this experiment for 100 days and make a histogram, right? So you all agree? So this is drift and mutation. That, okay, let's call that something, X. Frequency of A1, okay? This is going to be a random variable, RV. Okay, and I'm going to ask you, what's the distribution of X will look like? No calculation required. I want you to use these two facts that you learned. How deterministic populations, remember what I said in the beginning? How do we approach this problem? Just break it apart into small digestible pieces. I don't know what drift does, and selection and mutation, mutation and drift together do. But what I do understand is, if I turn off drift, I know what mutation does. If I turn off mutation, I know what drift does. Can I combine those two elementary lessons to get some idea about what happens when I have drift and mutation? So I would like you to think and feel free to discuss with each other, except Archana, because she knows the answer. Is the question clear? Probability. Is the question clear or not? Okay, I'll say it again. I said I ran this program. There's no selection. There's only drift and mutation. That means I have n individuals, and I am counting every day at t equal to, at some time. Every day I count how many A1s are there, and I make a histogram of that after a long time. So I started the program today, and I come tomorrow afternoon, and I just collect the data and plot a histogram. You all agree that there will be a histogram, right? That's the meaning of stochasticity, right? It's not a fixed number. So sometimes I'll see, in some cases, all have become A1. Sometimes there is all A2. Sometimes 3 A1. Sometimes 2 A1s, and so on, right? So what's the proportion like? What do you think? How many times would you see all A1s or all A2s or half A1s, half A2? Can you sort of come up with some idea about this? Let's think a bit, please. Yeah. Random variable. So population was very large. Let's think from that point of view. Population is very large, not infinite, but very large. How do you think this distribution looks like? X1 is a fraction. It has to lie between 0 and 1. Okay, right? Okay, so this much. And this probability, this also should lie between 0 and 1, right? Okay. So imagine I have a very large population. I'm not simulating 5 individuals, but a billion individuals. And there is this back and forth going on. So how many A1s do you expect to see? What's the fraction of A1s you expect to see tomorrow? What do you think? Half? Exactly half. Somewhere. Means what? It's a distribution, right? Come to the board and draw for me. Come, come. We like you guys. Come. That's next. Your turn. So be prepared. I'm going to call any of you. Let's think of very large populations. How do you think a distribution looks like?
Having like totally 0 A1 or all A2 is very less the problem, because this side is very less. Similarly, the other case is all A1 and 0 A2 is very less. So It should go something like this.
And where is it peaking?
At around
0.5?
and the frequency will
Does it make sense or not? Sorry? Yeah, mutation and drift, that's all. Yeah.
at this point mostly the highest probability is both are half. So 0.5.
So, but did you use the fact that you have a very large population? Suppose the population was small, would it still be the same?
Population is small. No.
Then what would it be? So, okay, maybe he is right. There's some probability that he is right. Okay, anybody? Do you think this is correct? I heard one yes and one no. Yeah, I mean, yeah, that's correct, yeah. So let's say one is over there. Yeah, no, you're right. But let's worry about the shape. Does this make sense?
This is under equal mutation.
Yes, yeah. Yeah, half, yeah. See, if you have understood something, so we should be able to, test ourselves. So that's what we're trying to do. Have I really learned something or not? No. Hold on. There's 35 people, other 35 people. I want to hear from them. Otherwise, I'm going to feel really depressed today. So, would you like to tell me whether this makes sense? Just tell me whether it's a sensible thing or not.
It depends on how often the thing is.
That's half probability. With half chance, I make A1 to A2 and A2 to A1. And we learned that if I have an infinitely large population, frequency of A1 and A2 would be half. But this is as a drift, yeah. Huh? No, I didn't say. I said n is million. It's finite, no? Let's first talk about the large population. Is this correct? Let's worry about this one. Large populations. Because large populations, we should be able to tell, right? Large population, million. Is it large enough? Meaning, what do you think is a good approximation? So this drift, but population size is very large. What is a good approximation to that case? Is drift important? So that's what you have to say loudly. No? Is drift important? Probably not. Oh. This is for large population?
Yeah.
Okay, so explain to me why is that?
I don't know exactly.
But I like this curve. No, I'm sure that's not what you mean. But
Not for a small population, it should be like this, but not for
okay. So she has this suggestion. This is the behavior for large population. Okay, so let's go ahead a bit. So the reason we do these deterministic models in which we neglect drift, they are a pretty good approximation and the population size is pretty large. Okay, so what is large? I told you, you can't just say, large and small. Large and small compared to something. Okay, so it turns out, which I have not shown you, that the relevant parameter here is, can you guess? No, suppose it was mu. S, there's no S, is neutral. What might be the relevant parameter? I told you in the morning. There's no migration. You have to adapt according to the question. N mu. So the parameter that is relevant is N mu. So if N is much, much larger than 1 by mu, then it's a pretty good approximation to treat it deterministically. Okay? And we learned that deterministic population is such that it has a frequency for mu equal to half at exactly half. But this is not exactly infinite population, right? So, like, you drew, it's more or less right. It sort of looks like this. Okay, good. Hold on. So this, more precisely, is valid when n mu is much, much larger than 1. So we have used this insight that we got from solving these unrealistic models. This is slightly more realistic, but we use that unrealistic models calculation to get to this. Okay? Yeah. Okay. Now, n mu less than 1. Does this make sense with everybody? For large populations, is more or less like a deterministic answer. Okay? That's how it looks. And I'm saying it will look like this because it's a calculation which has been done and we can't do this here, but the exact answer is known. Okay? So I know that this is correct. What happens with n mu less than 1? Is drift important there? Who is more important? Drift or mutation? Drift, right? So when n mu is smaller than 1, I can basically forget more or less about mutation, right? So this is more or less like the drift only problem. Okay, so that's what I meant. Switch this off, learn everything about it. Switch that off, learn everything about it, and then put on both of them. Okay, and put that information together. So now, if n mu is much smaller than 1, I can neglect more or less drift. So tell me, how would the distribution look like? I want somebody new to come. Who hasn't been to the board? Come. You're all very photogenic guys here. Want to come? Wow. Yes. Yeah.
So there, the probability that x1 is 0,
Yeah.
probability of encountering that frequency, like will the intercept actually be 1 minus?
I didn't understand.
The probability that you will
I don't know. This answer is known exactly. I'll come to that and then you can check that. Okay. So right now we're just doing sort of qualitative stuff. So this is the answer for n mu larger than 1 and n mu less than 1 looks something like this. Sort of a U-shaped function. There will be some teeny chance of getting half also, half A1s and half A2 because there's mutation, right? So there is this funny distribution. When I change n mu, it changes the shape so much. Click to U-shape. Okay? Right? Yeah?
Just trying to correct this to equilibrium. So,
oh, good. So when I said I will start a simulation today and check my frequency A1s tomorrow, this is what I meant. I meant the frequency in equilibrium at large times. So sorry, I should have said that. This is the equilibrium frequency. That means at large times. And that's why you could use half. So I should have said it, sorry. Okay.
correcting. So would you say that, for example, when nu is much larger than 1?
Yeah.
So 0.5 is the equilibrium point, because at that point if you're there,
Equilibrium and all with respect to time, no? Where's time in this? So frequency of x1. I'm making a histogram or rather continuous distribution now. I'm making a distribution now. Okay? Is okay? Yeah. That's the equilibrium distribution. Yeah, okay.
the peak will be at the point
Of the deterministic model, yeah. Yeah, that's right.
So in case of mu, say mu and mu other, that would be mu by
U plus nu. Yeah, that's correct. That's what you meant? Okay. Yeah? Okay, so now the math C, guys. Quick question. Do you know of any distribution which if you change a parameter below 1 and above 1, it changes the shape so drastically? Who said? See? It's valuable to have people with statistics background. Wonderful, that's correct. So this distribution is known, using some machinery, and it has a special name. It's called a beta distribution. Okay, and I hope I have a formula, full formula for it. Okay, I don't. Okay, but I'll write the answer. Yeah, why don't you tell me? So it's x1 into 1 minus x1. Is it right? To the power. In this case, it turns out to be 4 n mu minus 1. There's some proportionality constant, which is normalization constant. If it was not half-half, then you get a slightly different, the powers here, more generalized one. Okay? Okay, so this is called a beta distribution. Okay? Homework. Now, put in selection also. So I want you to think about this, and please do discuss with each other. If I now also have selection, so now A1 and A2 don't have equal fitness. One of them has slightly higher fitness, choose whichever you like. But I have mutation, selection, and drift. How does this distribution change? I'll ask tomorrow to come to the board and you have to answer it. Is it clear? Yeah, so the fitnesses are not equal now. Okay, so again the same funda. Turn off one of them and try to sort of put them together. It takes time, probably a whole night it will take, but you can live without sleep, can't you? Okay, fine, I'll stop here.